Hello everyone, myself I am Dr. Violet and today I am here to discuss the CSIR NET 2024 June questions and answers from Unit 4. So before I start, please let me know if I am loud and clear and if the slides are visible. Okay. Thank you students for confirming. So before we start with the discussion, let me give you an overview of Unit 4. So Unit 4 will have questions from host-parasite interaction. Okay, it will have questions from immunology, it will have questions from cancer, apoptosis, cell signaling, cell-cell communication. So these are the topics that comes under Unit 4. And first I'll be discussing the questions from the morning session. Okay, so again in that it will be like first part B questions and then we'll continue with part C. Similarly, once we are done with morning batch, means morning session, we will continue with evening session, where again first I'll be discussing questions from part B and then part C. So that is how we are going to discuss the questions from Unit 4. So let us start with the questions from the morning session first. So the first question number is 703532. As we can see, it is from immunology. So which one of the following statements regarding mammalian innate immunity is incorrect? 1. Pattern recognition receptors or PRRs are a component of innate immunity. 2. Serum complement proteins are part of innate immunity. 3rd, innate immunity has only a narrow range of specificity. 4th, the outcome of innate immunity is the rapid recognition and phagocytosis or destruction of the pathogen. Okay, so we have to find over here the incorrect statement. It is a part B question. So can you all try and tell me? So they're asking about innate immunity. So this is a very simple question. Let me if we talk about the standard of the question, difficulty of the question. So it is comparatively very simple, where you need to know the characteristic of innate immunity. So here the answer for this question, okay, so some students have answered right. So the right answer for this question, it is going to be option 3. Innate immunity has only a narrow range of specificity. This is incorrect. Okay. So why it's incorrect? So let us see the different statements. Pattern recognition receptors are a component of innate immunity. Definitely yes. So that is correct. Serum complement proteins are part of innate immunity. Yes, the serum proteins. Okay, the complement proteins, they are present in the serum and they are part of innate immunity. They are not a part of adaptive immunity. Okay, though they, they complement the antibodies, but still they are part of innate immunity. So second is also correct. 4th says the outcome of innate immunity is the rapid recognition and phagocytosis or destruction of the pathogen. So this is also correct because if we talk about the innate immunity cells like macrophages, okay, monocytes, so what they do is first they recognize and then they can phagocytose, right? They can destroy the pathogen. So 4th is also correct. Now 3rd, innate immunity has only a narrow range of specificity. Now this is not correct. Why? It has limited diversity, we can say. Okay, but it does not have a narrow range of specificity. It identifies some motifs, right? So for example, it can identify LPS. So LPS is present in a wide range of bacteria of different strains, right? So in that way, it is not identifying a narrow range. Okay, it does not have a narrow range of specificity. So that is why the option 3 or the statement 3 is an incorrect statement, right? So here we can see more in details about the pattern recognition receptors. So they are of different types. They can be membrane bound, they can be cytoplasmic, okay, they can be secreted. So we have different types, and in details, if you follow this table, you can see the membrane bound can be toll-like receptors or C-type lectin receptors. Again, toll-like receptors are of different types that will identify different ligands, okay, like as I was talking about lipopolysaccharide, peptidoglycans, flagellin. Now here the toll-like receptors, they are specific, okay, like according to the number, they are specific for certain ligands. But again, if you see, so like flagellin, it will be present in various bacteria that have flagella. So it is not that it has limited specificity, okay. Similarly, we see cytoplasmic, that is not like receptors, It can identify various ligands, okay, secreted at the mannan binding lectin. So they'll identify the sugar groups on the surface of microorganisms. So this table you all can go through in details and try to memorize this table, where you have the characteristics, you have the PRRs and the different types of PRRs and the ligands. Okay, so these innate immune responses are not specific to a particular pathogen. Okay, how it is specific for adaptive immune response. So that is why option 3 is not correct. They depend on a group of proteins and phagocytic cells that recognize certain conserved features of pathogens and become quickly activated and destroy the invaders. Okay, so that is why statement 3 is incorrect. So serum proteins, PRRs and macrophages, they are involved in phagocytosis. So they are all components of innate immunity. So now let's move on to the second question. So the second question is, again it's a part B question. Which one of the following statements correctly explains the function of a GTPase activating protein, that is gap in the regulation of heterotrimeric G proteins in plants? So what we have to find correctly explains. So gap, we have to see there, it activates G beta protein, it inactivates G alpha protein, it directly inhibits ligand binding to GPCRs, it leads to the dissociation of G alpha from G beta, G gamma subunits. Okay, can anybody try? Okay, so let us see the answer for this question. So correctly explain. So GTPase over here, what it correctly does in regulating the heterotrimeric G proteins in plants is it inactivates the G alpha protein. Okay, so let us see more in details about it. So if you see the GTPase activating proteins, how they function is by accelerating the GTP hydrolysis activity of G proteins, right? So this G protein has various subunits, has G alpha, gamma, beta, right? So in the context of heterotrimeric G proteins, gaps specifically act on the G alpha subunit. So here if you see, okay, so this is the G protein which is bound to GDP, it is an inactive state. Now GTP hydrolysis will take place. So that time what happens, the alpha gets separated from the beta and gamma. So this G beta gamma becomes active, okay, and G alpha bounds to GTP, that also becomes active. So gaps specifically act on the G alpha subunit. So when G alpha is bound to GTP, it is in an active form and it can transmit the signals downstream and further signaling will take place. So these gaps, they enhance the intrinsic GTPase activity of the G alpha subunit. So now what they will do is, because they have the GTPase activity, okay, so they will convert this GTP back to GDP. So now what will happen, this G alpha will become inactive. Okay, so what is the gap doing? It is actually inhibiting this G alpha, right? So it inactivates the G alpha protein. So once inactivated, this G alpha will now reassociate, okay, as you can see. So it's like a cyclic process, it will reassociate back with the G beta gamma subunits, returning the heterotrimeric G protein to its inactive state. So that is why the option 2 is correct over here. Okay, so this is again not a very complicated question, very simple if you know the characteristic about the GTPase activating proteins. And other statements if you see, so they are quite different. It activates G beta, so the G beta, that is not correct. It directly inhibits ligand binding, so that is not the function. It leads to dissociation of G alpha, so it is not dissociating. So once it inactivates, G alpha will again go and bind to it. So this part is again not correct. Okay, so the right answer is option 2. Now let's move on to question number 703534. Again, it's a part B question. So the question asks, which one of the following statements regarding Plasmodesmata in a plant cell is incorrect? Okay, so this is basically from cell-cell communication. And it's a simple question about Plasmodesmata. We all are familiar with Plasmodesmata. So first statement, they are specialized cell-to-cell junctions. Second, there are open channels that connect the cytosol of adjacent cells. Third, the plasma membranes of the adjacent cells extend continuously through each plasmodesma. Fourth, Plasmodesmata are extensions of chloroplast. that interconnects the cytosol of the adjacent cells. So it is quite simple. You have to just find the incorrect statement over here. So 1 will be incorrect. Quickly, can anybody solve and tell me? Okay, right. Yes, correct, students. So here the quite obvious incorrect statement is they are extensions of chloroplast. No, they are not extensions of chloroplast, right? It's written over here, they are extensions of chloroplast that interconnects. Definitely it connects the cytosol, so that is correct, but they are not extensions of chloroplast. So option 4 should be the right answer over here. Okay, so again we see it is a very simple question, not very difficult. I'm sure everybody might have solved this question. Okay, so let us see more in details over here. So you can see if these are two plant cells, so how their cytoplasm is connected. Okay, so the connection over here is the plasmodesma. Okay, it is not extension of the chloroplast. So it consists of a narrow thread of cytoplasm that passes through the cell walls, connecting the cytoplasms of adjacent cells. So this channel over here is lined by plasma membrane, ensuring continuity between the connected cells membranes. Okay, so it is not an extension of chloroplast, and other statements are correct. So they are cell to cell junctions. They are open channels, as we saw in the figure. It connects the cytosol of adjacent cells. This is also correct. The plasma membranes of adjacent cells extend continuously through each plasmodesma. So this is again correct over here. The plasma membranes are of the adjacent cells extend continuously. Okay, so the channel is lined by the plasma membrane. So it is lined by plasma membrane, okay, ensuring continuity between the connected cell membranes. So option 3 is also correct. So what is not correct over here is option 4. Now let's move on to the next question. (703589). So now we will be discussing the part C questions. Okay. So the first question is, which one of the following options represents all correct matches between column X and column Y? Okay, so we can say this question is from host-parasite interaction. Okay, or you can also say it can be from various diseases in immunology, where we study about some of the diseases like HIV, influenza, and then the question is about you have to match between the microorganism and the receptor, because when we discuss about diseases we also know to which receptor it goes and binds. Okay, so if you have an idea about the receptor and the microorganism, you can easily solve this question because it is simple. Again over here it is match the following type. Okay, so let us see if anybody can solve. I'll wait for some time. Okay, so let us see. So influenza virus, okay, to what it goes and binds in the host cells. Okay, so actually influenza virus, it goes and binds to the sialic acid residues of glycoproteins and glycolipids. Okay, so A matches with 3. So if we know A matches with 3, so the right answer should be option 4. Now let us see the other microorganisms and the receptors. Entamoeba histolytica. Okay, so B will actually match with N-acetyl glucosamine. Okay, Streptococcus pyogenes will match with CD44, and HIV we all know, so it goes and binds to CD4. Very simple. Okay, so here you can see if you know D4, okay, so you have two options 1 and 4. But again, influenza virus also is very simple, so we all know it. Sialic acid residues of glycoproteins and glycolipids, it goes and binds. So again, A matches with 3, so the right answer should be option 4. Okay, so these type of questions where it is match the following, so they are more scoring. You should try to solve more and more of this type of questions. Okay, so a little bit more in details if you see over here. So the figure is for HIV. So HIV goes and binds to the CD4 receptors, okay, on the CD4 cells, okay, the T helper cells. So they have the CD4 marker, as well as they need certain co-receptors. So you can see HIV needs certain co-receptors also, like CXCR4, CCR5. So once in, if you see previous year question paper, so they had asked questions related to their co-receptors as well. So you all should be well versed with the receptors and the co-receptors as well. So influenza virus, host receptors, sialic acid residues of glycoproteins and glycolipids. Okay, so the commonly known receptor molecule for influenza virus is a sialic acid. So it is also known as N-acetylnuraminic acid. Now this is covalently linked to the end of oligosaccharides on glycoproteins or glycolipids. So remember N-acetylnuraminic acid. That is the sialic acid. So Entamoeba histolytica we already saw, N-acetylglucosamine. Streptococcus pyogenes is CD44. So the CD44 is also a receptor for Streptococcus pyogenes, and where it colonizes basically is the pharynx region of the respiratory tract. Okay, and for HIV we already discussed. So host receptor is CD4, that is the T helper cells. So this is about the interactions between the microorganisms and the receptors. Next question is 703590. SLN1 receptor, a part of two-component system, is required for osmoregulation in yeast. The yeast mutant SLN1 lacking the SLN1 receptor protein dies. A researcher tries to rescue the mutant by expressing an Arabidopsis gene for the cytokinin receptor CRE1, which, like SLN1, is also a histidine kinase that acts through a two-component system. The following statements are made regarding the outcome of the experiment. A. SLN1 and CRE1 proteins respond to the same external signals, and therefore CRE1 rescues the yeast mutant. B. As CRE1 cannot interact with the downstream signaling pathway in the yeast, it will not rescue the yeast mutant. C. CRE1 will rescue the yeast mutant only if cytokinin is present. D. The vector domains of CRE1 and SLN1 proteins are sufficiently similar, and therefore CRE1 can induce the downstream signaling pathway and rescue the yeast mutant. Okay, so as we can see over here, they are talking about a two-component system. So it is related to the signaling part. Okay, in unit 4. So SL1 is required for osmoregulation. The yeast mutant lack this SL1 receptor. Okay, so that yeast mutant will die. So a researcher will try to rescue that mutant. So he will express an Arabidopsis gene for cytokinin receptor, that is CRE1. So this is like SLN1. It is also a histidine kinase that acts through a two-component system, and various statements they have given. That is the outcome of the experiment. So we have to find which of the following options represents the combination of all correct statements over here. Okay, so I'll give you all some time. Please try to solve and let me know. So if you read through the question, okay, if you see the question, at one particular glance it looks little complicated, but when you read and you understand what is happening in this question, okay, that they are doing some experiments, okay, and there are certain outcomes that we expect and some outcomes that we do not expect. So it is not very difficult, right? So let us see if anybody can solve. We will discuss this quickly. Okay, are you all able to solve this? Okay, some students are trying to solve. Let us see. Okay, so let us try to solve this question. The answer for this question, okay, it is option 2. That is, the combination of correct statements over here is C and D. So if you see the two component system, how it works. Okay, in the two component system, there is an input, there is output. Okay, so signal will come. So if it is if it's a one component regulator, so now the signal comes, that is the input, and then you get a response. So this is basically one component regulator. Okay, but if we could talk about the two component regulator, so in that case you have a kinase. Okay, you have an input and output. So a kinase is associated with the input protein, okay, which that receives the signal, and the output, okay, it is associated with the receiver protein, okay, that ultimately will give the response. So we have two components. One is the membrane linked kinase over here, the other one is the transcriptional regulator. So once the signal comes, there will be autophosphorylation of this protein, it will get activated, and because it has the kinase protein in it, so it will phosphorylate the receiver protein. Okay, so the receiver protein gets phosphorylated, which in turn will give rise to the response. So that is the output. Transcription will occur or not occur. So that is the regulator part. Okay, so we have two components, membrane link kinase over here and transcriptional regulator. Now let us see the different statements. So first statement says SLN1 and CRE1 proteins respond to the same external signals and therefore CRE1 rescues the yeast mutant. Okay, so in the mutant they have expressed this CRE1, but are they responding to the same external signals? A is incorrect, right? Because SLN1 and CRE1 do not respond to the same external signals. As written in the question itself, SLN1 is involved in osmoregulation in yeast, while CRE1 responds to cytokinin. Therefore, simply having CRE1 in the yeast will not necessarily rescue the mutant without the appropriate external signals. So, if they are saying that the mutant is being rescued, means it is not the input that is helping that. It will be definitely the output. Okay, so it is not necessarily just having that gene will rescue. Okay, you need the appropriate external signals, right? So, and they are not responding to the same external signal. So that is there. So statement A over here, okay, it is incorrect because one is responding to the cytokine receptor, other is required for osmoregulation. So they are not responding to the same external signals. Okay, so A is incorrect. B, it says as CRE1 cannot interact with the downstream signaling pathway in the yeast, it will not rescue the yeast mutant. So here B is again incorrect because the ability of CRE1 to rescue the yeast mutant depends on the similarity of the downstream signaling components. Okay, and not just on whether CRE1 interacts directly with the yeast signaling pathway. So if the signaling pathways can be successfully bridged, rescue is possible. Right? Are you understanding? So it is not because CRE1 can rescue, just because CRE1 and SL, they are responding to different signals. Okay, but B is incorrect because the ability of CRE1 to rescue the yeast depends on similarity of down. So what is the outcome? Okay, what is the downstream signaling components? So that will determine whether it can rescue or not. Okay, not just on whether CRE1 interacts directly with the yeast signaling pathway. So some successful bridging can occur, that depending on what is the outcome, rescue is possible. So that is why B is again incorrect, because it says CRE1 cannot interact with downstream signaling pathway. So it will not rescue the yeast mutant. Okay, so CRE1, if you see according to the question, it is helping, right? It is when it is being expressed, so the mutant is being rescued. So B is not correct, right? So B is incorrect. C, it says CRE1 will rescue the yeast mutant only if cytokinin is present. Yes. So here what is happening, because CRE1, the signal is different for both the genes. So CRE1 will rescue the yeast mutant only if cytokinin is present. So this is correct because in Arabidopsis, CRE1 is a cytokinin receptor. So for CRE1 to function and potentially rescue the yeast mutant, you need the cytokinin. Right, cytokinin has to be present. So the receptor CRE1 is specific to cytokinin. So its activity is dependent on the presence of this signal. So that is absolutely correct over here. It will rescue the yeast mutant only if cytokinin is present because it will only respond to that. Okay. So C is incorrect. Now D, it says the effector domains of CRE1 and SLN1 proteins are sufficiently similar and therefore CRE1 can induce the downstream signaling pathway and rescue the yeast mutant. So in the very beginning I told you all, what is important? Okay, if it is playing a role in rescuing the yeast mutant, means there is some similar downstream signaling that is occurring. Right, the output is happening same, okay, because of the downstream signaling. Only then it is able to rescue the yeast. So D is correct over here. Okay. So here the effector domains of CRE1 and SL1 proteins are sufficiently similar. Okay, it is correct because CRE1 and SL1 both operate through this two component signaling system. Okay, although they are from different organisms, if the downstream signaling components are compatible, CRE1 could potentially rescue the yeast mutant by inducing a similar signaling pathway. Clear? So that is why statement D is correct over here. So if you know the intricacies of how the signaling occurs, all the information is given in the question. You just need to apply whatever is written. Okay, it is analytical type of question. You don't have to memorize anything. You don't have to learn anything over here. Just that whatever is written, you have to understand this question and you can easily get to the right answer. So the right answer over here is option C and D. Okay, that is option 2. Statement C and D is correct over here. So that there is the question from signaling. So what do you all feel about this question? Was it simple, difficult? Yes. Yes, I agree that if you understand the question, then it's simple. Okay, but if you just overlook the question and you just you don't know about how this two component regulator works, so it might not be very simple. Okay, but if you understand the question, then it's a simple question that you can solve with little bit. If you read it carefully 2-3 times, you will get to the right answer. So let's move on to the next question. Okay, so this is question number 703591. So let us read this question. So here it's a graph based question, as we can see over here, and it's related to the NF kappa B signaling. Okay, so you can see this question is, if you know the signaling, okay, so you can easily solve the question. And the signaling, it can be a part of unit 4E also, because they are also signaling for the various cytokines is taught, as well as it can be a part of signaling chapter as well. So let us see the question. The inhibitory I kappa B alpha protein binds to NF kappa B dimer and holds them in an inactive state. On TNF treatment, the I kappa B alpha protein is degraded and NF kappa B enters the nucleus to transactivate gene expression. Importantly, one of the transcriptional targets of NF kappa B is the I kappa B alpha gene itself. Together, they form a negative feedback loop. Which one of the following graphs represents the NF kappa B protein expression kinetics when TNF exposure is given as a pulse or continuously? So A is when TNF is given as a pulse. Okay, B is when it is continuous TNF expression. So here actually you don't need anything about signaling. Okay, because everything is mentioned here. So what happens is, usually NF kappa B, it is a transcription factor, and it is present in the cytoplasm. Okay, if it is a cell, so it is present in the cytoplasm, it has to go inside the nucleus, okay, for transcription to occur. But it is bound by a inhibitory protein, that is I kappa B alpha, okay, or I kappa B you can say. So this I kappa B has to release the NF kappa B. So I kappa B gets phosphorylated, okay, when the particular cytokine comes and binds. Okay, so according to this question here, the cytokine is TNF. Okay, now there are two scenarios over here. One, either it is given pulse, means just once it is given, or it is continuous, the TNF expression is there. So which graph is most plausible or most correct. Okay, can you all try and solve this question? It's easy, nothing you have to, again, it's, you have to just see the graph and analyze it and tell what should be correct or incorrect. Okay, so let us see this question. So according to this question, okay, when TNF is given as a pulse, so what should happen? There should be NF-kappa B expression should be there, right? So NF-kappa B should become active, and then once TNF is over, its level means its activity will go down, right? Once TNF is removed, because again I-kappa B will be generated and then it will bind to NF-kappa B. So accordingly, the right graph for A part of the question, okay, should be this one. Okay, when the, so if you see 1 and 2, so they are same. Okay, so it can be 1 and 2. Now you have to see for the B also, when there is a continuous expression of TNF. So when there is continuously the TNF is there, so again what will happen? NF-kappa B will become active and again its level will go down, but again it will become active again. So it's like, it is continuously, its level will be altering. Okay, up and down. So that is why instead of 1, okay, the right graph should be option 2. Okay, when TNF is continuous, and here when TNF is given as a pulse. So if you see the signaling, so various stimuli, various cytokines will come and bind to the cytokine receptors, and you can see NF-kappa B is the transcription factor. It is present in the cytoplasm because it is bound to this I-kappa B. So because of this binding of the protein, certain signaling will occur. Okay, so the kinase becomes active, so it will phosphorylate this I-kappa B. So when I-kappa B becomes phosphorylated, it gets ubiquitinated and gets degraded. So proteasomal degradation occurs. So that is when NF-kappa B becomes active. Okay, it goes inside the nucleus and it acts as a transcription factor. So in pulse scenario, TNF is administered briefly, only once, right? It is for short duration. So leading to an initial activation of NF-kappa B. So this will trigger the degradation of I-kappa B, allowing NF-kappa B to enter the nucleus and leading to the expression of various target genes like the I-kappa B gene itself. So once I-kappa B is resynthesized, again it will go and bind to the NF-kappa B. Okay, so NF-kappa B will be again held in the cytoplasm. So there will be a rapid decrease in active NF-kappa B levels. So this results in a single peak. But when TNF is continuously present, the ongoing stimulation will cause repeated cycles of I-kappa B alpha degradation and resynthesis. So there will be kind of oscillations that are occurring in NF-kappa B activity. So we observe multiple peaks, not just one peak like this. Okay, so this won't be correct. So you will see multiple peaks. So as we see in option 2, the TNF pulse graph shows a single sharp peak consistent with transient activation. The TNF continuous graph shows multiple peaks. So it reflects the oscillatory behavior of NF-kappa B in response to sustained TNF exposure. Right? So the right answer is option 2. Now let's move on to the next question. So it's from cancer chapter. A cancer clinic is treating 4 unrelated patients suffering from chronic myelogenous leukemia. A researcher sequences the Philadelphia chromosome from the leukemic cells of these patients and makes the following statements. A. The DNA sequence was identical in the translocation breakage and rejoining sections in all leukemic cells in all 4 patients. So it is TBR they are calling, translocation breakage and rejoining sections. B. The DNA sequence was identical in all leukemic cells from patient 1, but every patient had a different TBR sequence. C. All patients have translocations between long arms of chromosome 9 and 22. D. All patients have translocations between long arm of chromosome 9 and short arm of chromosome 22. So which one of the following options represents a combination of all correct statements? So here you need to know about the CML, okay, why this disease is occurring, okay, chronic myelogenous leukemia, okay, so whether it is between long arms of chromosome 9 or long arm and short arm, so that is something that you need to know, okay, so this then only you will be able to solve this question. So you can try and tell me what is the right answer for this question. Okay. Okay. So, as some are mentioning, C is correct. Okay, let us see the right answer for this question. The right answer will be option 2. Okay, so yes, C somebody said correct. So C is correct. Okay, and so if C is correct, definitely D is not correct. And in A and B, so C, if you see the options, either it will go with A or B. So the right answer is B. So DNA sequence were identical in all leukemic cells from patient 1, but every patient had a different TBR sequence. So if you see, okay, the Philadelphia chromosome. So this is normal chromosome 9 and normal chromosome 22, which has the BCR gene. So when the chromosomes break, so it is between the long arm of chromosome 9 as well as the long arm of chromosome 22. Okay, so 9 and 22. So that is why C is correct over here. So it is change chromosome 9. So the exchange will occur. Okay, this green goes over here to the blue and blue comes here. So change chromosome 22 is the Philadelphia chromosome. Okay, where the BCR-ABL fusion gene occurs. So within a single patient, the Philadelphia chromosome is consistent across all leukemic cells, meaning the TBR sequence in the leukemic cells would be identical. Okay, so that is why B is correct. Okay, so the DNA sequence in all leukemic cells from patient 1, but every patient had a different TBR sequence. However, between different patients, the specific TBR sequence can vary. So this variability is due to differences in exact breakpoints. So wherever the exact breakpoint will occur, so depending on that, it might vary. Okay, where exact breakpoints occur on chromosome 9 and 22. So statement B is correct while A is incorrect. And CML is characterized by Philadelphia chromosome, resulting from a balanced translocation between the long arms of chromosome 9 and 22. So C is correct while D is incorrect. Okay. So again, this is not a very difficult question, but yes, you need to know about the Philadelphia chromosome properly and how it occurs. So let's move on to the next question, that is 703593. So if we see, it looks like this question is from immunology. So 100 IgM plus B cells were plated at 1 cell per well and activated in vitro. This led to detectable proliferation in all wells by day 4. At the end of 7 days, it was found that some wells contained IgG1 antibodies, some contained both IgG1 and IgA antibodies, and some contained only IgA antibodies. So what all we see? IgG1, then we see IgG1 plus IgA, and then some we see only IgA. So following interpretations were made. A. Cells that have switched to IgG1 may undergo further switching to IgA. Cells that have switched to IgA may undergo further switching to IgG1. A single cell can simultaneously secrete IgG1 and IgA. The progeny of proliferating cells may undergo independent switching events. So which one of the following options represents the combination of all correct statements? Now, if you have studied immunology thoroughly, you have studied about class switching. Okay, it is a very simple question because we know, okay, whatever the experiment, we don't have to follow also. Okay, if you know these statements, because in class switching, there are certain, steps how it occurs. So if you know those steps, it's easy to solve. So first thing, okay, the right answer for this question 703593, it will be option 1. A and D will be the correct statement because for this, you should know this is the direction in which class switching will occur. IgM, D, G, E, A. So from IgG, it can class switch to IgA. It can never happen that IgA will reverse. Okay, so if you know this part, then you know A is correct. Okay, IgG1 may undergo further switching to IgGA. So that is correct. IgA may undergo further switching to IgA. I said it can never reverse back. So B is incorrect. C says a single cell can simultaneously secrete. How will a single cell, if it is a B cell, it can either secrete IgG1, okay, another B cell can secrete, okay, another B cell can secrete IgA. It can never happen that one cell will secrete both antibodies, right? So that is, obviously, that will not happen because of, allelic exclusion. So C is again incorrect over here. D, the progeny of proliferating cells. So even if you don't know about D, you know A is correct. So A will match with D, and it is not matching with B. So it's very simple I would say. So the right answer is option 1, A and D should be the correct statement. The progeny of proliferating cells may undergo independent switching events. Yes, these are independent switching events that occurs. So D is correct. A and D. So the right answer is option 1. Okay. So class switching is the process whereby an activated B cell changes in antibody production from IgM to either IgA, G or E depending on the requirements of the cell. So choice of antibody will be influenced by the infecting pathogen as well as signals from surrounding immune cells. And it occurs via an irreversible process, that is class switch recombination, that you can see over here. Okay, it always occurs in this direction, never reverse. So this is what you need to know. Hence A is correct, B and C is incorrect, D is correct as it's an independent event. Okay. So now we come to the last question from the morning session. So let us see this question. A researcher was interested in detecting parasite-derived antigens in Plasmodium falciparum infected erythrocytes. The following labeling experiments were performed, followed by immunoprecipitation with antibodies against Plasmodium falciparum proteins and autoradiography. Okay, so these labeling were done. A, labeling with P32 ATP in media. B, labeling with iodine 125 in the media. C, labeling with S35 methionine in the media. D, labeling with H3 hypoxanthine in the media. Which one of the following options represents labeling experiments to predominantly detect the parasite-derived antigens? So if you have to identify parasite-derived antigens, okay, in the infected erythrocytes, so which labeling will be the choice? Okay, which labeling will be most appropriate? That is what is the question. Okay, you can try and tell me what should the right answer for this question. Okay, for this question I would say it is not, if you should have some idea if you are well versed with unit 13, okay, where we use different types of radioactive elements. So you can solve this question. Okay, so the right answer for this question, it will be option 2. Okay, C will be used. S35 methionine. Okay, so that will represent the labeling experiment to detect this parasite-derived antigens. So why it is like that? Let us see. So since mature erythrocytes do not incorporate amino acids, so when we use S35 methionine labeled proteins, okay, so they are synthesized by the parasite itself. Hence, labeling with S35 methionine will help in detecting the parasite-derived antigens because mature erythrocytes will not have it on their own. Okay. So, and they cannot incorporate the amino acids only when the parasite is there. Okay, so they will take up this methionine labeled proteins. Okay, that can synthesize the proteins. Okay, so that is how you can identify the parasite. Now let us see the other labelings. So if you label with P32 ATP, so it labels basically phosphorylated. So P32 means it will phosphorylate the proteins. Okay, so this is useful for studying phosphorylation events. It is not specific for detecting parasite-derived antigens unless you are specifically interested in phosphorylation state of these antigens. So only then we will use P32. B, labeling with iodine 125 in the medium is commonly used to label proteins through iodination. So it can label proteins from any source including parasite-derived proteins. So it can help in detecting parasite antigens. Okay, but it has to label the proteins through iodine. But here there is no iodination events that are occurring, right? So B is not correct. C, labeling with S35 methionine in the medium. So S35 methionine labels newly synthesized proteins by incorporating into their amino acids. So this is very effective method for labeling proteins specifically, including parasite-derived proteins, since methionine is an amino acid that is used by both host and parasite proteins. So it allows for a detection of newly synthesized proteins. So that is how the parasite can be easily identified. Now if we use tritiated hydrogen H3 hypoxanthine in the media, So this is used to label nucleic acids or metabolic intermediates. So Plasmodium falciparum incorporates hypoxanthine into its purine metabolism. Okay, so at that time it can be used for studying parasite metabolism, but it can directly cannot label the proteins. So if you're studying some metabolic pathway, so maybe that time you can utilize this. But if you have to identify the antigen, whether it is present or not, if you're detecting the disease, so for that purpose we need S35 methionine label proteins. Okay, so the right answer is only C, S35 methionine in the medium you should add, okay, to detect the parasite derived antigens. So that is all from the morning session. So if you analyze the morning session questions, so we see a lot of questions are asked from immunology. Okay, very simple, not very complicated. We saw one question from cancer. We saw one question as well from signaling. Okay, but at least we can say 2 questions in part C were from immunology. Okay, so immunology is important, as we can see this class switching question as well as this. Okay, this can also be, it can be a part of immunology, you can say. But because we can see the questions, okay, in unit 4 they have also been clubbed with unit 13, because this is immunoprecipitation and all that, it is a part of unit 13. But yes, you need to, as when we select the important units, so we always suggest to go for unit 4 as well as 13. Okay, so this is the reason why you should not skip these units, important units. So this is from cancer. This question again you can say it is from immunology. This is entirely from signaling 3590. Okay, and this is from unit 4A, host parasite interactions. Now let's move on to the next part, that is the question from evening session. So first again I'll be discussing first the part B questions. When E. coli and macrophages are placed in a petri dish with medium, the macrophages internalize the E. coli into cytoplasmic vesicles called phagosomes, which then fuse with lysosomes where the bacteria are killed. If E. coli is replaced by Mycobacterium tuberculosis in the petri dish, which one of the following options will happen after attachment of the bacteria? 1. The bacteria will be internalized by pinocytosis. 2. The bacteria will be internalized by autophagy. 3. The bacteria will prevent the maturation of phagosomes. 4. The bacteria will prevent the generation of lysosome in macrophages. This is simple. I expect everybody to answer quickly. It's a part B question. So I'm sure everybody knows what happens when Mycobacterium tuberculosis is added. Okay, what happens? Definitely, it gets phagocytosed. Okay, so but what will happen after that? So remember, it gets phagocytosed but does not get killed. Okay, how usually E. coli can be killed. Okay, when it fuses with the lysosomes, the phagolysosomes, it forms and then the bacteria gets killed. But for Mycobacterium tuberculosis, what happens is it will undergo phagocytosis, but the bacteria will prevent the maturation of phagosomes. So as a result, it will prevent its killing. Okay, so option 3 will be correct over here, to be more precise. Remember, it will prevent the maturation of phagosomes. So if you see this figure over here, okay, so once it is internalized, okay, through phagocytosis, so once inside phagosomes, this Mycobacterium tuberculosis will secrete enzyme like phosphatase, okay, and certain other enzyme like serine threonine kinase to prevent phagosome-lysosome fusion. Okay, so the mycobacterial phagosome, that this part, okay, the light yellow circle, so it is prevented from fusing with lysosomes because it secretes certain enzymes and there is active recruitment of host protein, okay, this coronin 1 TACO. So lysosomal associated membrane protein 1, that is LAMP1, and vacuolar type H plus ATPases, okay, that are present in the lysosomes. So they are components of the lysosome. So this Mycobacterium tuberculosis proteasome neutralizes the effect of nitric acid that is generated over here. within the macrophages. So nitric oxide tries to undergo the killing of the pathogen. Okay. So toll-like receptor signaling pathway will modulate the phagosome-lysosomal fusion via the activation of P38 MAPK pathway. Though it is not very clear whether mycobacteria interfere with this pathway. Okay, but definitely what it does, it inhibits the fusion of phagosomes with lysosomes. So that allows it to survive and replicate within the phagosomal compartment. So it avoids destruction by the lysosomal enzymes. So this strategy helps the bacterium evade the host immune response. So once inside phagosomes, Mycobacterium tuberculosis secretes the enzymes and the kinase, so that prevents the phagosome-lysosome fusion. So this is the exact step of what happens. Okay, it secretes enzymes, the phosphatases. So the correct answer is option 3, where the bacteria will prevent the maturation of phagosome. So phagosome cannot form the phagolysosome. Okay, so fusion of phagosomes with lysosomes get inhibited over here. So right answer is option 3. Next question. So we can say this question is from immunology. Okay, so this is a part of immunology question. Let's see the next part B question. Again, if we see, it is again from immunology. So you all should be well versed with immunology. In the thymus of a normal mouse, positive selection of T cells is based on recognition of which of the following? 1. Foreign antigens in association with self-MHC molecules. 2. Self-antigens in association with foreign MHC molecules. 3. Self-antigens in association with self-MHC molecules. 4. Foreign antigens in association with TLR ligands. Can you all try this? So it is based on selection, okay. So when the T cells, okay, they form CD4 and CD8 cells. So there are two selection that occurs, right, in the thymus, positive and negative selection. This part of the question is related to that. Yes, so somebody answered the right question. The right answer, for this question is option 3. So positive selection of T cells is based on self-antigens in association with self-MHC molecules. Okay, so let us see more in details over here, the selection. So the T cell precursor will first undergo rearrangement with TCR genes. It forms immature thymocyte, okay, and then it occurs positive selection. So in positive selection, it will be expressed by different MHC. Okay, the antigen will be expressed on the self-MHC, class 1 or class 2. So that is what is positive selection. So those T cells that can identify the antigens, okay, self-antigen along with self-MHC, they will be selected. Those that do not interact with MHC molecules, they will undergo apoptosis. Okay, so that is what is positive selection. So it occurs when double positive T cells bind cortical epithelial cells expressing class 1 or class 2 MHC plus self-peptides with a high enough affinity to get the survival signal. So these cells will get the survival signal who are selected. Then comes the negative selection occurs when this double positive T cells will bind to bone marrow derived APCs, that is macrophages or dendritic cells, that will express class 1 or class 2 MHC plus self-peptides. But they have high affinity. Okay, so those T cells that have high affinity, so they will undergo apoptosis. So that is negative selection. So here you have to note that selection occurs on self-peptides in the thymus. So MHC presents self-peptides in the absence of pathogen. So that is why option 3 is correct. First, why it is incorrect? Foreign antigens in association with self. No, it is not foreign antigens. So it is self-antigens. Again, self-antigen with foreign MHC. No, it is the self-MHC. Okay, so 1 is incorrect, 2 is incorrect. Fourth, let us see. Foreign antigen associated with TLR ligands. No, it is not in association with TLR ligands over here. Foreign antigens, that too, it is not foreign antigen, it is a self-antigen associated with MHC molecules, self-MHC. Okay, so fourth is again incorrect. So this is again simple, not very complicated. Next, 703034. So here it says normal human fibroblast cancer cells that originated in stem cells and fibroblasts transduced with HTERT were passaged for 35 generations. Southern blot analysis was performed using DNA from above cells using radio-labeled probes for telomeric sequences. Which one of the following band patterns will be observed in the autoradiogram? Okay, so for this you should have, it is actually from cancer chapter, you should have idea about the cancerous cells. Okay, so cancerous cells basically, their telomerase enzyme will be active. Okay, so accordingly if we see if telomerase is active, so the right answer for this question, it should be option 1. Right? So know that for cancer cells the telomerase gene is active. For normal cells it is not active. Okay? Southern blot analysis was performed using DNA from above cells for telomeric sequences. Alright, so here, because in normal cells telomerase enzyme is inactive, so there will be reduction of the telomeric end. But in cancer cells the telomerase is active. Okay, and in the case where the normal fibroblast is substituted with HTERT. HTERT is again the human telomerase reverse transcriptase gene. So if this normal fibroblast has this gene, means there will be more telomerase activity. Okay, more telomeres will be there. Hence, for the later two cases, we'll find a larger band than in the case where only normal fibroblast is present. Okay, I'm sure you all are understanding this question. So, HTRT is expressed only in cells and tissues which are positive for telomerase activity, that is the tumor cells and fetal cells. But HTRT is not detected in normal somatic cells that lack this telomerase activity. So, that is why in normal fibroblast, the band will be short. Okay. 7 to 9 kb bands in fibroblast. So, it will not be very large band. Okay. The expression will be less. And in cancer and HTRT, 18 to 20 kb bands. Okay. So, the right answer is option 1. And others are incorrect over here. So, that was from cancer. Now, let's move on to the next question. Again, it's a part B question. Okay, again, if you see, so it is again from immunology. So, how many from part B immunology we saw? Around this is the third question, right, for immunology. So, the immune recognition of self molecules is important for which of the following events? Initiation of B cell activation leading to antibody production. Promoting the differentiation of hematopoietic stem cells. Third, recombination of the T cell receptor. Fourth, activation of the NK cells of the innate immune cell. So, the immune recognition of self molecules is important for which of the following events? So, it's simple. Can anybody tell me the right answer? Why the immune cell should identify, they should have to know the self molecules. So, in which type of cells it is important? Yes. So, the right answer for this question, it's option 4. Okay. Activation of natural killer cells of the innate immune system. So, NK cells need, when they are developing, so they need to recognize the self molecules. Okay. Now, let's see the other statements. Initiation of B cell activation leading to antibody production. No, this is not correct. Okay. They don't need to be, for the initial activation. Okay, that is not needed. They have to identify the antigen. Second, promoting the differentiation. No, this is again incorrect. For differentiation, we don't need the self molecules. Third, recombination is not dependent on this self molecule or foreign molecule. It is depending on the various gene segments that come together. So, this is again incorrect. So, the right answer is option 4 over here. So, let us talk about the NK cells. So on NK cells, they undergo a maturation step where they recognize the self molecules that are constitutively expressed in steady state conditions. Constitutively expressed means they are always being expressed. So prototypical example of this process is the MHC class 1 induced NK cell education, it is known as. Okay. So it has been known that NK cells follow an education process. So here when they are identifying initially, when they undergo maturation, so they identify the self molecules. Okay, so that is known as education process, where they get educated to become both competent to recognize the missing self and they become also tolerant to the self protein, so that in future they don't identify the own self proteins. Okay, so this is what is education process that occurs during the development of the NK cells. Right? So if you see this figure, this is an NK cell. Okay, it has inhibitory receptor, stimulatory receptor. So when it binds to stimulatory ligand and inhibitory receptor, MHC class 1 molecule, it will identify the antigen. So it will try to protect. So transformation or infection if it is occurring, so it will kill the target cell where it misses the self recognition. Where it has induced self recognition, it has already, it knows that it's a self protein. When there is transformation or infection, so that time there will be killing the target cell. Okay, depending. If it knows that it has been educated and it knows it is the self protein, okay, so it will not identify the own self proteins. Clear? So that is why it undergoes a maturation step through the recognition of self molecules. So it will undergo no killing over here. It will protect the normal cell. Okay. But if there is infection, so that time it will do the killing. Okay, be clear about that. Where it can identify, okay, the target cell through the MHC. Next is question number 703089. Plants perceive effector molecules of a pathogen and mount a series of events that lead to the activation of a defense response. Following statements are made with respect to events that occur within a few minutes of the effector perception. A. Transient change in the ion permeability of the plasma membrane. B. Efflux of potassium and chloride ions from the cell. C. Influx of calcium and hydrogen ions into the cell. D. Influx of potassium and chloride ions into the cell. And efflux of calcium and hydrogen ions from the cell. So which one of the following options represents the combination of all correct statements? Now this question, because they have mentioned about pathogen, so I have considered in unit 4, but it can be a part of unit 6 as well. Okay, but let us see how to solve this question, so that is more important. 703089. So here it is talking about how the plant mount a series of events that lead to activation of defense response. So how the defense occurs in the plants. Okay, now for this question the right answer is option 1. So A, B and C it undergoes. Okay. So the perception of pathogen effectors triggers a rapid transient change in the ion permeability of the plasma membrane. So this is actually a factual question. You should know how the response occurs, defense response. So this change is crucial for initiating the early signaling events in the plant's defense response. As part of initial response, plants often experience an efflux of potassium. So they will try to remove the potassium and chloride ions from the cell. So that is why B is correct over here. Okay, so there is a flux of potassium and chloride ions from the cell. This loss of ions helps to create a hypertonic environment in the cell because ions are getting lost, so water will also get lost. So there is a hypertonic environment in the cell. So that is associated with decrease in turgor pressure. So turgor pressure decreases. So this is a part of signaling cascade. So that will lead to defense activation. Along with the release of potassium and chloride, there is intake or influx of calcium and hydrogen ions into the cell. Okay, so as a result, when calcium is entering, so it is a critical signaling molecule. So what will happen? Again, various pathways gets activated. So various defense pathways will get activated because this is important for signaling. It is a signaling molecule. Along with it, influx of hydrogen ion will contribute to the acidification of the cell's interior. So when interior becomes more acidified, so that also plays a role in defense signaling. Okay. So what we saw over here, influx of calcium and hydrogens, that is also correct. And change in the ion permeability. So A is also correct. Okay, there is no influx, so this is just the reverse statement. So D is not correct. A, B and C statements are correct over here. Okay. So now let's move on to the next question. 703090. Okay, so again if we see this question, it is from immunology. So what we see is a lot of questions are asked from immunology this time, right? I'm sure everybody had studied immunology. Usually what happens, every year they ask just one part C question and one part B, right, in immunology. But this time if you go through, so a lot of questions are asked. Okay, even if you go through morning session or evening session. So you have more than one question from immunology. So this is a part C question. Let us see. The following statements are made about how CD4 T cells provide help to CD8 T cells. A. Antigen MHC2 complexes on CD4 T cells interact with antigen MHC1 complexes on CD8 cells. B. A single dendritic cell presents antigen on MHC1 and MHC2 at the same time. C. CD4 T cells activate dendritic cells which produce chemokines like CCL3 and CCL4 that can specifically attract CD8 T cells to form a CD4 CD8 DC triad. D. CD4 T cells help B cells which differentiate into plasma cells and secrete antibodies that form immune complexes which bind to Fc gamma RS on CD8 T cells. So the question is, which one of the following options represents a combination of all correct statements? Right? So can you all try and tell me what is the right answer over here? For this question, you should be well versed about CD4 T cells. Okay, how they get activated, what are the different MHC they require. Okay, what are the different APCs that activate them. So a lot of things that you need to know about the MHC molecules, you need to know about the various T cells. Okay, their markers, whether they are CD4, CD8 cells. So you need to know a lot of information about the T cells, APCs, MHC. Okay. Not both are correct. So let us see. Yeah, B is correct. There is one more statement, right? You have to find the correct statement. Okay, for this question 3090, the right answer is option 3. Okay, B and C statements are correct over here. So let us see this statement A. Antigen MHC2 complex on CD4 T cells interact with antigen MHC1 complex on CD8 cells. So T helper will have CD4. Okay, so they identify the antigen on APC that will have MHC class 2 with the antigen, right? And it will bind to the T cell receptor. Correct? So does it interact with antigen MHC1 complex on CD8 cells? No, right? So A is not correct over here. B, a single dendritic cells present antigens on MHC1 and MHC2 at the same time? Yes. So dendritic cells can express both MHC1 and MHC2. Okay. So B is correct over here. C, CD4 T cells activate dendritic cells which produce chemokines. Okay, so these CD4 T cells can activate dendritic cells, and yes, they can produce certain chemokine like CCL3, CCL4 that in turn will activate the CD8 T cells. So they form a CD4 CD8 DC triad. So C is correct. D, CD4 T cells help B cells which differentiate into plasma cells and secrete antibodies that form immune complexes which bind to this receptor. Again, D is not correct. Okay, they do help B cells by releasing cytokines. Okay, but they don't go and bind to Fc gamma because of the CD4 T cells. Right, so D is not correct. So what is correct over here is B and C. Okay, so if you see these dendritic cells, they express both MHC class 1 and MHC class 2. Okay, so MHC class 1 will activate CD8 TC cells. MHC class 2 will activate CD4 T helper cells. Okay, so that is there. So dendritic cells which initially activate helper T cells as well as targets of effector helper T cells such as macrophages and B cells. Because dendritic cells express both MHC class 1 and class 2, they can activate both cytotoxic and helper T cells. So B is correct. Dendritic cells are activated by CD40L expressing CD4 plus T cells act as mediators of T helper signals for CD8 plus T lymphocytes. So as I said, so first they'll get activated, they'll release various cytokines. So inducing their cytotoxic function. So further they'll activate the CD8 T cells. So C is also correct. Now T cell receptor MHC interactions alone do not efficiently trigger T cells. So it requires the participation of co-receptors, that is CD4 and CD8. So because of that, there are different T cell subsets. So CD4 is expressed on Th1, Th2, Th17 helper cells as well as on CD4 T regulatory cells. CD8 is expressed on CTLs and CD8 T regulatory cells. So CD4 and CD8 enhance T cell signaling by binding MHC class 2 or MHC class 1 molecules on APCs. So that is why A is incorrect. Okay, and T cells do not have antibodies receptor. So how D will be correct? Okay, so D is also incorrect. So they do not have antibody receptor as written over here. They do release cytokines to activate the plasma cells and they can secrete antibodies. Okay, then that form immune complexes that bind to Fc on CD8. No, they do not have the receptors present on T cells. So again, D is incorrect. Okay. So this is, I would say the difficulty level of this question is medium, not very simple, not very difficult though. Okay, but if you have some knowledge about how, about all these cells, MHC, APCs, okay, T cells and specific dendritic cells, so then you can easily solve this question. And let us move on to the question number 703092. The following statements are made regarding formulation of advanced candidate anti-malaria vaccine, RTS. This is from vaccines. A, it contains heat-kill Plasmodium falciparum sporozoites and hepatitis B surface antigen. B, it contains formalin inactivated Plasmodium falciparum sporozoites and attenuated poliovirus. C, it contains a fusion protein between Plasmodium falciparum, CSP, C-terminal region, hepatitis B surface antigen. D, it contains a fusion protein between Plasmodium falciparum merozoite, surface protein and CSP. Now this is from immunology. So here in specific you should know about this antimalarial vaccine, okay, how it is formulated, okay, RT3. Okay, so for this question let me tell you all the combination of all correct statements. The right answer over here is option 4. So how this antimalarial vaccine RTS is formulated is, it has a fusion protein between Plasmodium falciparum, CSP, C-terminal region, hepatitis B surface antigen. Okay, so remember RTS is a recombinant protein based vaccine. It contains a fusion protein between the C-terminal region of the circumsporozoid protein of the Plasmodium falciparum as well as the hepatitis B surface antigen, that is HBsAg. Okay, so you can see in details. So this is the HBsAg. Okay, and the CSP amino acids, C-terminal amino acids. So there is a fusion protein between the C-terminal region of this circumsporozoite protein and the hepatitis B surface antigen. So primary vaccination will include 3 doses. Booster dose is given after 20 months. So what is the immune response over here? So various antibodies like IgG, IgA, IgM will be released. T cell help antibody production as well as memory B cell generation. CD4 plus T cells will play an important role as well as CD8 plus T cells. So various modifiers like age, nutrition, so all that will also depend on the immune responses. Complement fixation can occur, complement proteins will get activated. Okay, so the receptor for the antibodies will further, so Fc gamma receptor binding will occur and various immune cells, neutrophils, monocytes, NK cells will become active. Okay, so here you can see the vaccine efficacy in children. What is the efficacy? So it is around 35%, young infants around 20%. Okay, with the booster doses. If booster dose is not given, so it is still less. So efficacy is less. Okay, and this is the kinetics. Okay, for the antibody level. So primary response, what is happening? Okay, and here is the booster dose that is given to maintain the antibody levels. And let's move on to the next question. So this is from cancer. A cancer cell line obtained from a rat glioma tumor was stained with the nuclear dye HAX 33342 and sorted using FACS. About 0.4% of the population stained lightly, distinct from the densely stained population of cells. Equal number of cells from these two populations were subcutaneously implanted into a suitable animal model to develop tumors. Following statements are made from this experiment. The LSP cells will give rise to tumors. The DSP cells will give rise to tumors. The LSP cells can give rise to LSP and DSP cells. The DSP cells can give rise to LSP and DSP cells. So which one of the following options represents the combination of all correct statements over here? Okay, so here you need to have some idea about how once you are staining the cell, the cancerous cells, how they take up the stain. So if you know that, you can solve this question. Okay, so this experiment described. So the right answer, as you can see, it is option 2, that is A and C. So LSP cells will give rise to tumors and LSP cells can give rise to LSP as well as DSP cells. So the experiment described involves sorting this rat glioma tumor cell line using FACS. So lightly stained cells are typically thought to represent a subpopulation with stem-like or cancer stem-like properties, while the densely stained will represent more differentiated cells. Okay, remember that. So here, based on the properties, the LSP cells will give rise to tumors. So this is true because LSP cells, they have cancer stem-like properties. So they are capable of initiating tumors. So that is why A is correct over here. DSP cell, it says give rise to tumors. So this is likely false because they are more differentiated. So they have a lower or no tumor initiating capability compared to LSP. So B is incorrect. Okay, C, the LSP can give rise to LSP and DSP cells. So this is also likely true because these are stem cell like properties they'll have. So they can differentiate into various cell types. So again, it can be LSP, it can be DSP. So C can be considered correct. D, the DSP cells can give rise to LSP and DSP. No, because as we said, DSP is already differentiated. It is more differentiated. So they are not capable of reverting back and going back to the stem-like state to give rise to again LSP because they are already DSP. Okay, so D is incorrect. So as we can see over here, option 2 will be the right answer. Right? Now we come to the last question from evening session. So the following statements are made about the killing of virus-infected respiratory epithelial cells by cytotoxic T cells. So again, if you see, this question is from immunology. A, priming of the T cells has taken place in thymus, lymph node or spleen. B, viral antigens have been presented on infected epithelial cells. C, MHC1 molecules have been presented on infected epithelial cells. D, MHC2 molecules have been presented on infected epithelial cells. So which one of the following options represents the combination of all correct statements? So if you carefully go through, the question is simple, it is not difficult. Okay, so when we study immunology, we know how the virus infects, which type of MHC is expressed on which cell. So if it is a virus infected respiratory epithelial cells they are talking about. So the virus has infected the epithelial cell. So what should happen? Can you quickly tell me the right answer for this question? Okay. Yes. So the right answer for this question, it is going to be option 1. Because it's very simple. Okay, viral antigens have been present. Definitely the antigen, where it will be, where once the virus enters these epithelial cells, so the antigen will be presented on the infected epithelium. They'll take over the machinery of the cell and they'll start expressing their own protein. So the viral antigens will be presented. Right, so B is correct. C, MHC1 molecules have been, definitely the antigen will be presented on an MHC1 molecule. Okay, so C is also correct, so that it can activate the CD8 T cells, okay, or the TC cells, correct. So B and C are correct. Now D is talking about MHC2, so that is not happening, so D is not correct. Priming of the T cells have taken place in thymus, lymph node or spleen. Now again, this is not a correct statement, okay, because when the development is occurring, so the naive T cells, they leave the thymus and then they migrate to the secondary lymphoid organ. So that is where they are primed and differentiate into effector cells with certain specialized phenotypes. So again, A is incorrect over here. Okay, because here it is mentioned that priming takes place in thymus, so this is incorrect. And other statements already we went through. So epithelial cells infected with live influenza virus effectively present viral antigens and were lysed by both homotypic and heterotypic influenza virus specific CD8 plus T cells. So B and C is correct while D is incorrect. So if you see the, how the priming is taking place. So once these T cells, they develop in the bone marrow, they go to thymus, okay, they form different cells, CD4, CD8 cells, and then they go to the periphery. Okay. So that is all from Unit 4. Now I pass on the controls to the teachers who will be discussing the questions from Unit 5. Thank you everyone for being attentive. Thank you.
Good evening everyone. Yes, am I audible? Please confirm in the comment section in the chat box. Yes, very good evening to all students present here. So, okay, my voice is clear. Thank you for confirming. Okay, my slide is also visible, right? Alright, great. So, today I am here to discuss about the Unit 5 topics, whatever questions were there in June 2024 CSIR NET exam. Okay. Now, since this Unit 5 is development biology, I will be discussing the questions which are on animal, based on animal development. Just after this, once I finish, Shamrita ma'am will be taking up the plant development biology questions. Okay. So let's begin. Before going into forward, let me give you a small analysis of this paper. So first of all, I'd like to know how did you find this particular unit? Unit 5, Development Biology. How did you find? I want to hear from all of you. Students who appeared for morning session, evening session. Both the questions we will be discussing today. All the Part B and Part C questions, like how we are doing it for all the other units. But I want to know, you can just write in the comment section whether you appeared for morning batch or for evening batch. And what did you feel? See, as such, this overall exam, the question pattern is not that easy. The complexity has increased definitely. Questions are no more straightforward. But when it comes to Unit 5, there are two very important observations. So, 2-3 important observations. Let me see if anyone has noticed them or not in the question paper. Yes. Yeah, it was tough, isn't it? Yes. Definitely the questions were not of easy level, I will say. Right. And Yes, experimental. Very good, very good. Yes, that's right. Experimental questions were there. Okay, so from my point of analysis, from on behalf of Biotechnica's analysis, what I can say is that this time the experimental based questions were more. Okay, and that too, the experiments did not cover on any one concept. They were based on 2-3 concepts. When I come to the especially the evening batch questions. So I think on in part C question 2, 3, 4 questions were there, 4-5 questions on animal. So out of that 2 to 3 were completely on experiments only. Okay. And another thing is that in part B there was 2 plant based questions. Generally they will give 4:1 ratio or 3:1 ratio in part B. But in evening batch there were only 2 animal based questions and 2 were from the plant based questions. Okay, and apart from that, the part C questions, they were experimental, but actually they were not like lengthwise it was not so much big. They were pretty summarized statements, just that some conditions or experiments were given and you have to get the answers. On the other hand, part B questions were also experimental. So as we will see in the morning batch, there was a Drosophila question. It was a diagram based question which came in part B. Generally, we expect the diagram based or table based everything to be in part C. Those are the analytical question. But actually here in this time, unit 5, you got the other way around. Okay, it happens for some other units also. Anyways, let's move forward. First, we will be discussing the part B questions, morning sessions. The first question was actually a combination with unit 2, I would say. In animal cells, typically which organelle is only provided by the sperm to the oocyte following fertilization? Okay, so here the keyword, as you know in CSIR, the keywords are important. Here the keyword is only, right, which is not contributed by the oocyte. We all know that sperm contributes the nucleus, isn't it? But they have asked which organelle is only provided by the sperm to the oocyte. So that is, oocyte will also have nucleus. If you look at the zygote, then nucleus is provided both by the sperm and the oocyte. So that's why it will not be the answer. The nucleus or nucleolus will certainly not be the answer. And we also know that the mitochondria and flagella, even if it is given to the oocyte, then anyways they will disintegrate. Okay. And we know that in sperms, when sperms are made, most of the organelles are not present. The cell undergoes a process of differentiation, because of which the other organelles are removed from there. The Golgi complex is modified into an acrosome and the other organelles are not present. So peroxisome cannot be answer. The correct answer is centrioles. Alright. So in most mammalian reproductive systems except for rodents, the sperm contributes a centriole to the fertilized oocyte that duplicates during the pronuclear stage and separates from after syngamy to serve as mitotic centers. So what happens is, whatever the centrioles are there in some of the organisms, it will duplicate, and from there some asters or microtubules or they will be radiated out. So on one hand, those will be contacting the male pronucleus and the female pronucleus, like in sea urchin only it happens, and it would be pulling them together. Okay, so the sperm is the sole contributor of centrioles to the zygote, and these centrioles are thought to be essential for fertility also. Sperm centriole recruits maternal proteins and forms a large aster associated with the sperm inside the zygote. Okay, that was pretty simple actually. Now the next question, it was based on cortical reaction in sea urchin. As such, sea urchin is not mentioned in your CSI syllabus, but still most of the times questions come from this particular topic. Which one of the following statements is correct? Okay, so one is entry of calcium ions into the egg initiates development, right? So entry of calcium, it comes in the egg. This part is not correct because calcium is already stored inside the cell endoplasmic reticulum, and from there it is released into the cytosol. So calcium doesn't really come inside the egg. Yes, the calcium release into the cytosol from the endoplasmic reticulum store is definitely important for continuing the development. That is how the further development processes will begin, but the entry of calcium is not correct. This part is not correct. That's why. So you have to select the correct statements. So one cannot be answered. Exocytosed cortical granules during egg maturation contain the components of the zona pellucida. So what we have is the cortical granules are present when you have the cortical granules just below the plasma membrane. When this sperm fertilizes the egg, when one sperm fertilizes the egg, this cortical granule contents are released towards the outside. Okay, and outside the plasma membrane, so this is the plasma membrane under which the cortical granules are present, and outside the plasma membrane we have the extra membrane layer which is called as zona pellucida. This is present in mammals. Okay, so zona pellucida is penetrated by the sperm. Okay, the acrosomal reaction, whatever sperm acrosomal contents are there, they will be released, and that is how the sperm can enter the zona pellucida, and then only it can fuse with the membrane of the egg. Okay, but here they are saying that the cortical granules contains components of zona pellucida. How is that possible? Zona pellucida is a membrane, isn't it? It's a layer, glycoprotein membrane. So glycoprotein layer rather, not membrane, it's a glycoprotein layer. So how can cortical granules contain the components? Okay, what cortical granules actually contain is for some components which can convert the zona pellucida into fertilization envelope. Okay, because once one sperm is fertilizing the egg, other sperm should not be able to fertilize the egg. That is why this particular layer has to be hardened by the components which are released from the cortical granules. So from zona pellucida it will become fertilization envelope. Okay, that is why the component 4th is correct. See what happens? The release of the cortical granules after sperm entry, some mucopolysaccharides are released from here, from the cortical granules, and sugar molecules means they attract water. By taking water inside, this zona pellucida gets uplifted little bit. Okay, and the hyaline deposition also takes place, and there is some hydrogen, some peroxides are also released which can harden the fertilization envelope. So this is what is correct. It can convert zona pellucida also and vitelline envelope also. So since we are talking of sea urchin, so instead of zona pellucida you can consider vitelline envelope. Zona pellucida is in mammals, and in lower animals, amphibians, sea urchin, so there you will have vitelline envelope. Okay, now the depolarization, that's why 4th statement is correct. The depolarization of the plasma membrane after entry helps to block the polyspermy. This sentence as such it is correct, but the question is asking about cortical reaction. It is not asking about the fast block. This is a part of the block to polyspermy which happens immediately after fertilization, which is called as the fast block. Fast block is achieved by depolarization, not by cortical reaction. Cortical reaction is a component of the slow block process, and that is why, since question is about cortical reaction, this can be a trap question for some people. They would not read what is actually asked, but they will just find out if the statement is correct or incorrect without even referring to the original body of the question. Okay, so in that case you will mark it correct probably, but you should also see what the question is asking about. Okay, so this was a tricky question in that form. So calcium ions, they are responsible for the cortical granule reaction and are stored in the endoplasmic reticulum of the egg. It does not enter the egg. Okay, and you can see here from the cortical reactions we have some peroxides which are released. You can see peroxides which are released. So initially the vitelline envelope, what was a thin layer, now you can see it has converted into a hard fertilization envelope. Okay, cortical granule reaction is a slower mechanical block to polyspermy, and mucopolysaccharide released by the cortical granule produce an osmotic gradient that causes water to rush into the space. So when the water is coming in this space, this gets slightly lifted up from the membrane, and that also makes it impossible for any other sperm to come and bind with the membrane. Okay, so it causes the envelope to expand and become the fertilization envelope. So one of the protein released by cortical granule is peroxidase enzyme. This is responsible for hardening the fertilization envelope by cross-linking the tyrosine residues on adjacent proteins. Fine. So when we are analyzing these questions and looking at the answers, you are getting to know many other information. For example, this you can take away for your next exam. What if in the next exam some question regarding this concept, they can ask that, they can ask tyrosine residues cross-linking. Based on that, they can ask a question or a statement. Alright. Moving on to the next question. This is based on C. elegans. In Caenorhabditis elegans, blastomere identity occurs through both conditional and autonomous modes of specification. So, which of the following options is a correct statement in this regard, right? So, these are the statements which are given over here. Now, in C. elegans, we know that we begin with something called as a P0, that is the embryo. And then this P1 will divide into two more cells. So, basically the embryo will have now two cells. It's an asymmetrical division in which you will have a bigger AB cell and then you have something called as P1 cell, okay? And then whatever you have, this AB cell and the P1 cell that you are having, they will be undergoing a different type of division. One will be dividing horizontally and the other will divide vertically, okay? So that way, when the cell is making two daughter cells, so one will be like this, one will be like this, and another like this, another like this, okay? But because of space adjustments, this will shift little bit here and this will shift little bit here. So ultimately, you are getting four cells. One is here, one is here, one is here, and one is here. Now these cells, the last cell is the P2 cell, which gives signal to these two daughter cells of AB. Okay, so now both are daughter cells of AB, but whichever is connection with the P2, that receives the signal, okay, to form ABP. And whichever doesn't receive the signal, that will become ABA. So this is the concept based on which they have asked the question. If the AB and P1 blastomeres are experimentally separated, now P1 is this one and AB is this one. If they are separated means the AB cell will generate all cells it would normally make. So AB cell is supposed to differentiate into ABP and ABA, but ABP will be done only while getting signal from one of the daughter cell of P1. So if you separate then AB cell cannot generate the cell. That is why this is incorrect. When AB divides to form daughter cells, ABP becomes different from ABA. So that is right, because when it is forming, AB is forming the daughter cell, ABP will be becoming different by receiving the interaction from the P2 through its interaction with P2. Okay. The specification of AB cell is determined by the presence of cytoplasmic determinant. See, cytoplasmic determinants, they are present inside the cell, because of which a cell will take up a particular fate. It doesn't need any signal from outside. But ABP requires signal from P2. So why will it need signal from P2 to become ABP if it already has cytoplasmic determinant? So this also cannot be correct. And P2 cell produces a morphogen for the determination of AB cell. This is also not correct, because P2, if it would have made a morphogen as we know, it generally gives a gradient, isn't it? And that too, it can influence more than one cell. But here, the P2 is influencing only the cell which is just next to it. It is not influencing the cell which is also next to it. This cell it is not influencing. So that's why it cannot produce a morphogen. Alright. So that's why the correct answer over here is option 2. If you look here, the signal that P2 has, P2 gives signal something which is called as APX1. So again, as I told you, when you analyze the paper, when you get to know the answers of the paper, you can take a lot of other informations for the next chapter, means for the next exam, isn't it? Or anyways, if you want to learn everything from scratch, then that's even more better. Obviously, with this you will get only limited information, but yes, this is like additional topics if you are not aware. Okay, so APX1, which is a delta type of signal. We have delta notch signaling pathway, which is very important from CSIR point of view. So APX1 is the signal name which is released from P2, and AB, whichever AB cell gets that signal, APX, only that cell becomes ABP. So the descendants accordingly will come, the other cell fates, okay? And the receptor on this AB cell is what is called as the notch, that is a GLP1. Okay, and that will be receiving the signal. Anyways, notch is there on ABA also. So even if you interchange their position, it won't matter. whatever will receive the signal, that will form ABP. So receptor is there in both, but because signal is just next to the P2, that is why only ABP will be receiving the signal. And that too, if you know something about Notch delta signaling, they are part of juxtacrine signaling. Okay, and juxtacrine signal means they need cell to cell contact. Right, so P2, if it was a morphogen, it would have released signal to ABA also. But since it's a juxtacrine type of signaling, only the cell which it is in direct contact, only that cell will be influenced. Okay, so you can understand how much intricate detail based on that they are giving questions these days. C. elegans demonstrates both the conditional and autonomous mode of specification. Both modes can be seen actually in the first two blastomeres, if the blastomeres are experimentally separated. So P1 can develop autonomously. P1, even if you separate from the cells, then also P1 will be making the cells what it is supposed to make. Okay, it will develop autonomously without the presence of AB. It makes all cells that it would normally make, and the result is the posterior half of the embryo. So this is because of cytoplasmic determinants. They already have it inside them. AB is not by cytoplasmic determinant. But however, the AB cell, if you do it in isolation, it will only make a fraction of the cell types that it would normally make. So thus option 1 is wrong. Therefore, the specification of the AB blastomere is conditional and it needs the descendants of the P1 cell to interact with it. Okay. The P2 cell is critical in giving a signal through a delta-like protein APX that distinguishes ABP from its sister. So option 4 is incorrect because it says morphogen. ABP becomes different from ABA through its interaction with the P2 cell. So option 2 is correct, right? Now this was the question in morning batch which I was talking about. It is a diagram based question. They have given some sketches of wild type and mutants. So this is the wild type diagram and they have given 3 mutant, first, second and third mutant embryos of Drosophila melanogaster. Which one of the following options represent a correct match between the gene and its loss of function phenotype? See, these mutations have occurred because of the loss of some genes, loss of function of some genes. So the question is asking about the loss of function only. Like which gene, if it's mutant, is giving rise to 1, or which gene is mutant is giving rise to 2 or 3. So first of all, look at the wild type. You can see some anterior, this axis they have also given, means this side is anterior, this side is posterior, and this side is ventral, this side is dorsal. So you can see towards the anterior dorsal side, you can see 2 head appendages which are present. Obviously towards the anterior, you can expect the head to be present. But you can see that in one, the head development hasn't occurred that well. And we know that in Drosophila, bicoid controls the head development, okay, and nanos will control the like posterior development. Alright, so these are the maternal genes which are there, but even before that, there are some more genes which are involved in the anterior, posterior and dorsalization and ventralization. So that time only some problem might have been there, because anyways none of these are mentioned over here. Okay, so we know that the dorsal identity of the Drosophila, it is defined by something called as gherkin. Okay, like gherkin, if it gives a signal to torpedo, torpedo is present in the ovary and gherkin is released from the embryo. Okay, because of that torpedo, pi protein is not synthesized, and that is why dorsal protein gets free. Okay, dorsal protein gets free, and like dorsal protein is not free because cactus is bound with it, and that is why it cannot enter the nucleus towards the dorsal side. It's an entire long sequence. I am not going into that sequence. So you can see that here, if the dorsalization would have happened properly, then you would have got the head appendages. But clearly the dorsalization has not done properly over here. Here it is completely absent. The dorsalization is completely absent, and here something else has been formed. So dorsalizing signal from the oocyte nucleus appears to be the product of the gherkin gene. As I said, if gherkin is there, so I'm not going to detail of that entire sequence. So just remember that if gherkin is there, then the oocyte gets dorsalizing signal and it will be towards this side. So you can see some dorsal appendages are there. Now mutation of the gherkin gene. If you have mutation of gherkin gene in the mother, okay, and that is why the mutation will be there in oocyte also, then dorsalization will not happen. That is exactly what has happened in this one. Okay, there is no such there is a mutation because of which there are no dorsal appendages. It causes ventralization of both the embryo and also the surrounding follicular cells. So those cells will lack the dorsal structure and appendages which are present in wild type. So sketch 2 represents this type of embryo, which means it represents a gherkin mutant. That's why the correct answer will be option 2. So if gherkin is not there, then only the complete embryo will be ventralized. Okay. So let's move on to the next question. It is regarding if chimeric mouse embryos were generated using GFP expressing embryonic stem cell. So GFP is green fluorescent protein and RFP red fluorescent protein. That means chimeric means 2 different cells were generated, means 2 different cell types. So one are the embryonic stem cells and another one is the pluripotent stem cells. Okay, the different colors are so that you can identify them. And basically they have taken embryonic stem cells, okay, and they have taken induced pluripotent stem cells. So anyways, we know both are pluripotent in nature, so they can give rise to all the cells of the body. They can make all the cells of the body. Which one of the following tissues from any such resulting embryo? If you are making embryo taking these stem cells, then they should be able to produce any cells of the body. Okay. So which of the following tissues from any resulting embryo will not express any fluorescent protein? So if these embryos are raised into proper animals, then which of the organ will not contain any fluorescence? So not contain any fluorescence means those cells will be made from other source, not from these sources. Okay, so that means which is that organ which is not made from embryonic stem cell and induced pluripotent stem cell. Now as we know, in the mammalian blastula, we have, once the blastula is made, we have a layer of cells which are present outside. Those are the trophoblast cells, and some cells are present inside, which are the inner cell mass. Okay. Now we know that the outside trophoblast cells, they can produce the extra embryonic layer, mainly the placenta, and the inner cell mass, they are composed of the embryonic stem cells. Okay, so they are able to make all type of cells. And that's why if you take a chimeric embryo containing embryonic stem cells and induced pluripotent, they will be able to produce all the cells of the embryo proper, except for placenta. And that is why these cells are pluripotent. Okay, if they were able to make the all the cells, like even the placenta also, then we would have called it totipotent. But they are not totipotent, they are pluripotent in nature because they cannot make placental components. Okay, so you will not see any fluorescence in the placenta because these are not derived from these cells. And these cells, whatever is derived from these, those will have the fluorescence in them. Whatever part is not derived from these, that will lack the fluorescence. So they will not contain that. They will not express that fluorescence protein. So correct answer is option 4. Embryonic stem cells and induced pluripotent stem cell can both contribute to the formation of embryo itself. So all the other things which are mentioned, that brain, heart, intestine, they will be able to form those. These cells can differentiate into the 3 germ layers, ectoderm, mesoderm and endoderm, which form the body tissues including the brain. They will from ectoderm, heart from mesoderm and intestine from endoderm. However, the placenta is primarily derived from the trophoblast cell, which are part of the outer cell mass of the blastocyst, and they are not derived from the inner cell mass. Okay, so that forms the embryo proper. So ESCs and IPSCs contribute to the inner cell mass, but do not contribute to the trophoblast lineage. Therefore, in chimeric embryos generated using GFP expressing ESC, all embryonic tissues would express, but the placenta will not express, right? So that was, those were all the questions of morning batch part B. I would like to inform you all about the Josh batch, which is for CSIR NET December 2024 and 2025 exam. You can enroll to this by calling us on our toll-free number, which is given here, or you can also call the office number. These are the components that you get along with this Josh batch. Okay. If you are enrolling now, seats are limited. So in case if anyone is interested in clearing the exam, this upcoming exam, then you must hurry up with the enrollments, because this offer is limited, where you are getting so many things at one go. Okay. you can talk to our student support team and definitely they will help you out, right? So yes, moving on to the part B questions of evening session. How many were from evening batch? So morning batch students, did you find this helpful? Anyone of you could clarify why your question went wrong? Okay, so let's move forward to evening batch questions. Which one of the following statements best describes an acrosomal reaction? It is a repulsive interaction between the sperm and egg. It involves digestion of the acrosome by the sperm when it encounters an egg. Okay, so we are talking of the acrosomal reaction from where some particles from some things would be released out from the acrosome reaction, some enzyme and some components. From the acrosome itself, it will be released. So how can it cause digestion of acrosome? That is not possible, isn't it? And it's because of this acrosome reaction only that the egg membrane is penetrated so that the sperm can go and fuse with the egg membrane. So definitely it is not a repulsive interaction. It leads to the digestion of the zona pellucida. Yes, this seems a correct option, because the outer layer, what is there in mammalian system, when the sperm binds to the zona pellucida, it undergoes acrosome reaction and then it will be releasing the contents, because of which it can enter this perivitelline space. Here it will be going and fusing with the egg membrane. Okay, so the acrosome is already removed. Acrosomal reaction at the zona pellucida is already happening. So it is not involved in the fusion process. Okay, for fusion, there are plasma membrane fusogenic proteins, they will be helping, but not the acrosome reaction. So third option is the right answer. Okay, if you see the diagram over here, when a sperm cell makes contact with the zona pellucida, it undergoes morphological changes which are termed as the acrosomal reaction. The acrosome reaction also leads to a release and activation of acrosine. It is a proteolytic enzyme that is most likely one of the enzymes involved in the digestion of zona pellucida, necessary to facilitate the sperm transport of sperm into the perivitelline space. Okay, so you can see here that the acrosome, once it is binding with the zona pellucida, this is zona pellucida. Okay, it is releasing the contents and then it comes in the perivitelline space, and then it will fuse with the membrane of the oocyte. Okay, so it has nothing to do, acrosome reaction is happening before fusion of membrane only, and anyways this is done by fusogenic protein, not by acrosomal membrane. So this was also a pretty simple question. Then another question, mutations in a specific signaling pathway result in early defects which are observed in establishment or maintenance of midline structure. So midline structure means in the body, in the midline we have the vertebrate column. The primitive structure is what is notochord. Later notochord forms vertebrate column, and also the floor plate. So this is done by a particular signaling pathway. It is very important. And if there is a defect in that particular pathway, then you can see defect in these structures as well. Later defects include absence of distal limb structures, ventral cell types within neural tube, spinal column, and most of the ribs, and even cyclopia, where there is a single eye located at the center of the face. So this, all of these places, this particular signal is very important, as we know in limb structure also. So if anyone would have done a decent Unit 5 preparation of either neurulation or limb development or eye induction, then they would know that the pathway which is in concern is sonic hedgehog. So the SHH signaling pathway, SHH gives the signal for dorsalization also, then for limb also, limb development, eye field formation, and also the limb structures as well. Okay, so mutations in which of the following signaling pathway is the most reported cause for these congenital defects? So answer would be option 1. Okay, SHH signaling cascade plays an essential role in patterning the dorsoventral axis of the neural tube. So as you can see, this is the neural tube, and here we have the ectoderm, and this is the notochord, right? So when this is formed, SHH in notochord gives the signal to neural tube to form the ventralization. So complete absence of SHH signal, as in null mutant, results in a dramatic effect on the dorsal ventral patterning. If SHH signal is not there, then dorsal ventral patterning is severely affected, Because the SHH is giving rise to the ventral side of neural tube, that is why it will lead to absence of ventral cell type, and also the floor plate formation will not be able to form properly. As you can see, this is the floor plate. Okay, only if SHH is there, then only the floor plate will form, else it will not form properly. Eye fields are also abnormally close together or fused in the midline. This is a condition which is called as cyclopia. Okay, so that was all. As I said, in part B questions, in evening session part B questions were less. So only 2 questions were there. And rest were part C question. Rest were the plant-based questions. Okay, so now coming to the part C questions, first we'll discuss for the morning session. The following statements have been made regarding cell specification in an early embryo. So they have given a diagram in which there is a ball of cells. From here, certain cells are removed through glass needle. But yet, if you see the outcome, you can see a normal development which is formed. So this is an amphibian embryo, looks like an amphibian embryo, which is kind of tadpole structure, which is kind of normal only, right? So even if you're removing some cells, still a normal embryo is formed, means if again you have a decent preparation, then you would know that this is talking about conditional specification. So to some extent, you can keep that in mind and progress with the rest of the questions. The following statements have been made regarding cell specification in the early embryo. The entire embryo rarely interacts with its environment, and its development trajectory cannot be guided by its immediate ecosystem. Okay, so if you are talking about conditional, then entire embryo rarely interacts with its environment is not correct, because in conditional the cells get signal from the nearby cells, and that is how they will develop properly. So they have to interact with the environment. So development trajectory cannot be guided by ecosystem. This is wrong. So A definitely cannot be answered. Okay, so this option you can remove off. So you have to see whether it is C, D and B. When cells are removed, changes in cellular biochemistry and function are never preceded by a process involving the commitment of the cell to a certain fate. So when cells are removed, you can expect that there will be some changes. You can expect in conditional that there will be some changes in the cell fate. Okay, cell fate will be changed. Otherwise, how they are coming back to normal? Okay, so some cells would have compensated. They would have changed their fate. Then only they are committing to, then only their fate is changed. Otherwise, whatever these cells were supposed to give rise to, they would have gone missing in the embryo, as it happens in autonomous specification, right? So if these are not present, you can expect some cell fates to change, right? The fate of a cell or a tissue in the intact embryo is said to be specific when it is not capable of differentiating autonomously on being placed in a neutral environment, such as petri dish or test tube. So if you are if a fate of a cell, if it is specified, then even if you take that cell out and culture it separately without the rest of the embryo, if it has already been specified, if it has already got the signals, then they will be able to differentiate by themselves, even if the other cells are not there in a neutral environment. Okay, but it is saying the other way around, that it is not capable. That's why C is wrong. So if C is wrong, anyways B was wrong. As I said, if you remove the cells, then some cells will change their fate to compensate for the missing cell. So B is wrong, C is wrong, only D is the right answer. So anyways, in conditional specification, if cells are removed, the interaction of the remaining cells compensate for the fate of the removed cells, because the fate of a cell depends on the conditions in which the cell finds itself. So the only way out here is they will make more and more cells over here, and by making more cells, these cells will change their fate according to what cells are present next to them. That is why any structures is not seen missing in this part, and a normal embryo develops this way. Okay. So this is the explanation what we generally have. This is what we study in the classes, that the remaining cells can regulate and compensate for the missing part. Okay. This is directly relevant to the experiment where normal larva development occurs despite cell removal, right? So a few questions in the morning batch, they were very easy actually. They were direct questions, but some were a little bit twisted. For example, here is one more such example which is little bit twisted. The homozygous mouse homozygous null mutant for a particular gene. So gene name they have given BFG. The mouse homozygous null mutant means BFG is not there. then the mouse always dies in mid-gestation. It means complete development is not able to take place. But when you take chimeric mice, and chimeric mice which are made up of BFG null cells plus wild type cells also. So if you are taking an embryo in which you have some BFG null cells, but you are taking along that some wild type cells, then embryo is able to survive. But if you are taking an embryo which is having BFG null cells only, then that cell is never able to survive. Okay, so better to solve such kind of questions with a diagrammatic representation. When the developing brain of these pups is examined, so the brain is examined, they have more than 2 hippocampus. These healthy pups who are able to survive, they have more hippocampus. Okay, more hippocampus is present. Now, closer examination reveals BFG null cells at the center of this supernumerary campus. So whatever this hippocampus is there, many hippocampus is formed, right? So in the center you are finding some cells which has BFG inside them. Okay, so which of the following influences can be correctly drawn from this experiment? This is only the experiment result. Now we have to fit in the results over here. So the first option says that wild type BFG function in the developing brain induces hippocampus specification in neighboring cells. See, whatever hippocampus is there, the center cells in the center of the supernumerary hippocampus, you have BFG null cells, right? So if BFG would have induced hippocampus formation, then you cannot expect at the center the BFG null cells to be there, isn't it? Then if BFG was responsible for induction, in the center BFG null cell wouldn't have seen so many hippocampus nearby. Yeah, you got it. See, what I'm saying is you have the supernumerary hippocampus formed from this embryo, isn't it? In the center you are getting a cell which doesn't have BFG. So if BFG function was to induce the hippocampus, how can you find the so many hippocampus around a BFG null cell? If this was inducing, means you wouldn't have seen so many hippocampus, isn't it? If BFG is not there, then hippocampus should also be not there. But you are seeing that even if BFG is not there, hippocampus is present in lot of amounts, isn't it? So this option cannot be correct. Second option, on the other hand, does make sense because wild type BFG function in the developing brain suppresses hippocampus specification. That makes sense because BFG suppresses, doesn't allow to form hippocampus. Okay, that is why if BFG is not present, okay, so it cannot suppress the hippocampus. That is why so many hippocampus is formed. Okay, wild type BFG function suppresses the hippocampus. BFG function is completely cell autonomous. So this also we cannot say is correct because if BFG function were completely cell autonomous, the lack of BFG in certain cells. So when we have taken BFG null cell, it would have affected only those cells and it wouldn't have influenced the neighboring cells. But since the absence of BFG in BFG null cells affects the surrounding wild type cells, because of which extra hippocampus is formed, so we can say that BFG is giving out some kind of signal. That is why it is able to suppress the hippocampus. So it cannot be cell autonomous. Okay. And BFG function is only hippocampus specific. So again, this also cannot be completely supported because the experiment suggests that BFG plays a role in hippocampus development, but it does not provide enough evidence to conclude that BFG function is exclusively hippocampus specific. So anyways, they have not asked here that what is factually correct. Otherwise, you could have told 4 can be an option. But this question is not about factual correctness. It is about what inferences you can draw only from this experiment. So since this experiment is not providing any results or any observation about the other things which BFG can do, so we cannot definitely conclude about this. But this can be definitely concluded because if BFG null cell is there, if BFG is not there, that is why they are not able to suppress the hippocampus surrounding it, because of which the supernumerary hippocampus is developing surrounding it. Okay. So yes, this was a kind of twisted question and it needed some deep analysis. Anyways, the next question is not that difficult. In a hypothetical organism, a 4-celled embryonic stage, at a 4-cell embryonic stage. Now if anyone realizes this is nothing but combination of the C. elegans development what they have given. Okay, so anyways we will not compare it with C. elegans, let's solve it individually. At a 4-cell stage embryonic stage, blastomere X instructs one of the daughter cells of an adjoining blastomere Y. So X instructs the Y to take the fate YA. Okay, so okay, all right, you are not able to see. Yes, I will change it. Okay, I think now it will be clear to you. So X gives a signal to Y. Okay, and yeah, and the fate that Y is taking, sorry, so X is giving a signal to the Y. Okay, so it is this way round. So this is the signaling molecule and this is the receiving molecule. Now Y, if it is giving the signal, if it is getting the signal, it is taking up two fates. You can see you have a YA and YP. So as you can see, the cell which is getting the X signal, that side it is forming YA, right? So the cell whichever is X is instructing the Y to take up the fate YA. So X is giving the signal, Y is dividing into two daughter cells and X is giving signal to form the YA. Alright, and whichever cell is not receiving the signal, it is forming YP. So as we just solved the question on P2 and AB blastomere, it's like that only, but they have reversed the positions. So let's not bring that in between. The other daughter cell takes the YP. This is illustrated in the figure below as lineages of YA and YP. If the X blastomere is removed, both daughter cells take up the Y fate. Okay, YP fate, posterior. Okay, so if X is removed in the absence of blastomere, you can see that Y, both the Ys, it has divided in this lineage. You don't see this lineage over here. It has divided in this lineage, which means that both of these daughter cells have taken the posterior fate. Both of the daughter cells of Y. If the X blastomere is removed, both daughter cells take up the YP fate. This instruction is mediated by a paracrine factor. So why this signaling happens? It is because of a paracrine factor PAP2. So PAP2 is secreted from X, okay, interacting with PAP5. So they interact with this PAP5 over here, okay. So even there also we have in C. elegans, P2 gives signal to EMS daughter cell. Whichever cell it gets the signal, it forms the endoderm. So there also we have this MOM2 and MOM5. Anyways, this is hypothetical organism. So here they have named it as PAP2 and PAP5 secreted by X blastomere interacting with PAP5 on the Y blastomere. So following experimental manipulations were carried out which involved creating partially genetically mosaic embryos in vitro and following the fate of the Y blastomere. Okay. So which of the mosaics will show a development pattern similar to that when X blastomere is removed? So they have done, see this is one experiment what they have done, but the question is some more experiments are done and which experiment will give the same result like this. Okay, similar to that pattern when X blastomere is removed. Now for this, let's consider the normal development here. Okay, and based on that we will see what experiment result we are getting. So one is X blastomere null for PAP2. PAP2 is the signal. So if X blastomere doesn't have the signal, then you will not get the YP, isn't it? You will not get the YA. We know that X was supposed to give signal. So if the cell is not there, what effect will be there? The same effect will be there if the signal is not there. So PAP2 signal is not there, but you have taken wild type blastomere, but if X signal PAP2 is not there, YA cannot be produced. So that will give a similar signal to this one. Let's consider the other ones also. Wild type X blastomere. X is fine, it will give signal PAP2 and constitutively activated PAP5 blastomere. Okay, so constitutive activated means the PAP5 is always activated in Y. Now PAP5 is activated in Y means both the daughter cells of Y will have PAP5 signaling in them. They need not be activated by PAP2. The receptor is activated by itself. So signal is not required. In that case, both the daughter cells, they will go towards YA fate. Okay, which is not similar to this because in this case both the daughter cells are going towards YP fate. So that's why this won't be the correct answer. X blastomere none for PAP2. So X blastomere in this experiment, X blastomere PAP2 is not there and constitutively activated PAP5. Now here, as I told you, if you have constitutively activated PAP5, anyways both the daughter cells will have activated PAP5. So both of them will be taking YA. Whichever has PAP5 activated, it will form YA. Otherwise it will form YP. But here, since the Y cell itself is constitutively activated for PAP5, both the daughter cells will produce YA. Okay. So this is again not same with this one. Fourth is X blastomere null for PAP5. Again signal is not there. So if signal is not there, but you have taken wild type blastomere. So first of all, X blastomere none for PAP5. PAP5 is not the signal, it is the receptor. Okay. So whether X has the PAP5 or not, it doesn't even matter. Anyways, the signal is given by PAP2. So if PAP2, as long as PAP2 is there in X, it will be able to give the signal to Y because Y is wild type over here, and Y will be able to generate YA with the signal from PAP2. So that's why here also it will generate YA and YP, which is again not similar to this because this is YP and YP. So option 1 is the right answer. Okay. So you can go through this later in the recording. If it is still not clear to you, I'll have to go a little fast now so that we can cover all the questions. Now this was another question from morning session only. Overexpression of protein A in the brain of Drosophila melanogaster causes the degradation of ovaries in the animal. Okay. So if you have protein A overexpressed, then the ovaries are getting degraded. This is our observation. Overexpression of a secretion incompetent allele of A. Secretion incompetent means this cannot be secreted out. Even if it is present, it cannot be secreted out. So it is secretion incompetent allele does not cause this phenotype. Okay, so as long as it is not, A is not getting secreted, ovaries are still intact. Okay, ovaries are intact if A is no secretion. Okay, but if you are overexpressing A, okay, so no secretion means ovaries are fine, but you're overexpressing A, secretion is also there, then ovaries are getting damaged, right? However, down regulation of protein B in ovaries. In the ovaries, if you have B down regulated, concomitant with overexpression of protein A, means along with protein A overexpression, if you make in ovaries B protein come down, overexpression of protein A in the brain, it prevents ovary degradation. Okay, so that means alone expression of protein A in the brain, it damages ovaries. But if you have downgrade of B along with overexpression of protein A, it is not able to cause the degradation of ovaries. So you can say another one that A downregulation and A upregulation, B downregulation, ovaries are able to be present. Okay. So A and B are found to be physically interact with ovary lysets. Physically interact in ovary lysets. That means A and B are physically interacting with each other. Right? Physically they seem to form a bonding together. Right? So A and B, you should remember this. Basically they are asking something regarding signal. In the light of the above experiment, which of the following inferences could be correct? So protein A cell autonomously influences ovary development while B is secreted to influence the brain function. So have a look at this. The fact that a secretion incompetent allele of A does not cause the phenotype suggests that A must be secreted from the brain to exert its effect, isn't it? It is said that if you are having no secretion from A, then ovaries are still fine. So protein A cell autonomously influences ovary development, autonomously, and B is secreted. So A, when it is not secreted, okay, option 1 cannot be correct because it is mentioning autonomous. And we are seeing that secretion incompetent. That means secretion means it's part of a signaling process. Okay. The prevention of ovary degradation by downregulation of B in the ovary indicates that B is required for A. Okay, so that is why to cause ovary degradation. That's why when B is down regulated, then it is able to cause the ovary degradation. Thus, option 4 cannot be correct as it mentions that both molecules are released from ovaries, right? So it is said A is a receptor secreted from ovary and B is a ligand in the ovary cell membrane, right? So that's why it cannot be correct. The interaction between A and B, as I said, it suggests that B is a receptor for the secreted protein A, as signals directly interact with membrane receptor. So that's why option 2 is the right option, because it says A is a ligand secreted from the brain and B is a receptor in the ovaries. Okay, so that is why when A is getting signaled, it binds with the B. Okay, and that is why it will lead to ovary degradation, right? Ovary degradation. But what happens if B is down regulated? Then even if A is upregulated, the signaling cannot happen. That is why the ovaries will still be present. Okay, and if A is not secreted, okay, if in case you are taking B but overexpression of A, but they are not secreted, in that case also they will not be able to bind with B. In that case also ovary will be present. But in case the A is present high and B is not there, B is not down regulated, B is normally present, so overexpression of A will be able to bind with B and it is giving rise to ovary degradation. Okay, so it's a kind of a negative influence of the signaling process. Third is also not correct because A is a neurotransmitter secreted from the brain and B is a signal transducer. If you see the signaling pathway, the signal transducers are somewhere over here which bring about the result inside the cell, and the neurotransmitters will be outside the cell. So those never directly interact with signal transducers inside the cell. The signals will interact with the receptors, okay, but not with the signal transducers inside. But since here they are forming a lysate, neurotransmitters and signal transducers do not interact directly. But since it is mentioned that A and B interact physically in ovary lysate, thus option 3 also cannot be correct. Okay. So yeah, these are kind of tricky questions. And in case you want to get a proper understanding of the concepts so that you can solve such difficult questions, you will have to practice more and more questions. I would like to remind you regarding the Josh batch. Everything is covered in this, especially the question AI powered Innate test series, ToughNet question banks, and also the concept table, concept wheel. Everything is made available to you. Okay. However, you will be getting free access to the online study e-portal. Here all the recorded sessions are present. You get 24/7 chat support for academic doubt resolution, isn't it? Then we have the part C questions for evening session. The first question was from Drosophila. Wild type Drosophila have a pair of wings of one segment and a pair of halteres on the adjacent posterior segment. We know inside bodies are divided into segments. So in the thorax segment, there are 3 thorax segments from which one pair of leg comes out from each segment, and only from the second segment one pair of wings come out, and in the third segment some haltere is present. Okay. Now this particular segment is controlled by a homeotic gene which is called as UBX. Okay, and that is the third segment is controlled by UBX. Okay, so if UBX is not there, then halteres cannot form, and because of which the Drosophila gives rise to 4 wings. Not only in the second segment but also in the third segment it generates a pair of wings. So on the other hand, dragonflies, if you look, dragonflies naturally they have 4 wings only, right? Drosophila is supposed to have only 2 wings, right, from the second segment, but the third segment which is controlled by UBX, if it is mutant, then even in the third segment you get 4 wings, right? So UBX mutants of Drosophila have 2 pair of wings and no halteres. In relation to UBX function, for 2-wing structure species, insect species, and 4-wing structure species, they differ based on what? This is the question, what is all about. Okay. So if you look at the options over here, in Drosophila, UBX is expressed at a certain level in the third thoracic segment to repress the wing formation and promote the haltere development. The reason why normal Drosophila wing don't develop in third segment is because UBX will suppress the wing development. UBX is not there in second segment. That is why it is not able to suppress the wing development. That is why from second segment wing is present. However, this same function, that is repression of wing formation, is not seen in four-winged structure, four-winged insects like dragonflies, where the third thoracic segment also forms wings. So critical difference is not the amount of UBX, but rather how the presence of UBX leads to different outcomes, that is wings versus halteres, which relates more to the targets of UBX rather than its expression level. That's why option 1 can be removed, which says that UBX expression levels, it's talking about the amount of UBX that give rise to wing and haltere. As I said, it has got nothing to do with the amount. Okay. Now, while timing of gene expression can be crucial in some cases of development, the difference between two-winged and four-winged insects is not due to when UBX is expressed, but it is how UBX influences segment identity. So, option 2 also cannot be correct because it is talking about the timing. UBX regulation at different development times in segment that gives rise to wings and halteres. Okay. Then we have difference in UBX copy number of paralog evolution could theoretically influence segment identity if these copies had different functions. However, in this case, the difference between two-winged and four-winged insect is more about how UBX is functioning within the content of the segments where it is expressed. So, option 4, this would be option 4. Option 4 also can be ruled out because in option 4 it is saying that UBX copy number and paralog evolution can be answered. But third option is correct because the fundamental difference between the two-winged and four-winged likely lies in how UBX regulates the expression of genes downstream, which determine whether a segment forms halteres or wings. Differences in these downstream targets between species could explain why Drosophila has halteres and why dragonflies have additional wings. Okay, so since option 3 says target of UBX in segments that give rise to wings and halteres, that's where the differences are coming. Okay. Now the next question was regarding FGF expression in the anterior developing mouse brain, which induces anterior identity marker expression. That means FGF expression in anterior causes anteriorization. But FGF8 receptor, the signal receptor, it is uniformly expressed in the brain. So some of the experiments were done, these experiments. Which one of the following experiment best demonstrates this fact? So if you are expressing transgenic overexpression of FG8 in the posterior of the developing brain, right? So if FG8 is overexpressed in the posterior, then this will lead to anteriorization in the both posterior and anterior ends of the brain. So it would demonstrate that FGF8 is uniformly expressed and capable of responding to FGF8 regardless of the region. So this experiment directly tests the effect of FGF when ectopically expressed and supports the hypothesis that receptor is uniformly distributed. Okay, so that is why option 1 is only the right answer. However, the other options, let's see. Transgenic overexpression of FGF8 in the posterior in the developing mouse brain cause anteriorization at both ends of the brain. So option 2 would only demonstrate the effect of overexpressing receptor itself, not FGF8. It doesn't directly test the ability of FGF8 to induce anteriorization when expressed in different brain region. Okay. So grafting the anterior portion of a FGF null developing mouse brain into the posterior of another developing mouse brain causes the recipient's brain to be anteriorized at both ends. Option 3 experiment involves a FGF8, that is non-functional FGF8, thus doesn't directly involve testing the expression. Okay, and option 4, transgenic overexpression of FGF8 in the posterior results in no anteriorization. So anyways, this experiment suggests that FGF8 receptor is not uniformly expressed because posterior doesn't respond to FGF8. Okay, it is written that it does not give rise to anteriorization, which means that the posterior is not responding to FGF8. But the idea of this experiment should be to show that uniformly it is expressed. Then in that case, option 4 also should have given anteriorization. Okay. So however, this contradicts the idea of uniform receptor expression. So this was completely a logical question. There is no theoretical relevance as such. You have to solve it logically only. Next question was from the Xenopus embryo. Following experimental manipulations were carried out. One was where the embryo was exposed to ultraviolet radiation, because of which cortical rotation didn't happen. Let me tell you that cortical rotation is an important mechanism in Xenopus embryo, which is very important for the dorsal ventral specification. Because of cortical rotation, some components get aligned or accommodated in this area, which gives rise to the dorsalization of a structure. Okay, so if these are manipulated, cortical rotation is not there, then dorsal ventral specification will not happen. Towards the dorsal side, we have the organizer, which releases components like chordin and noggin. These are all dorsalizing agents which can stop the ventralization at this part. So if cortical rotation is not there, these all will not be able to dorsalize the particular structure. So here we have the manipulation X. Cortical rotation is essential for establishing dorsal ventral axis. The failure of cortical rotation generally results in a ventralized embryo with a loss of dorsal structures. Okay. Then manipulation Y, gastrule are treated with lithium chloride. Lithium chloride is an agonist of canonical WNT signaling. Okay. A very important component of cortical rotation, as I said, it is beta catenin. Okay. Beta catenin has to be accommodated towards the dorsal side so that the dorsalization can take place. But however, lithium chloride, if it is there, beta catenin not only accumulates in a specific region, but it accumulates in the entire embryo. Okay, and wherever beta catenin is there, it will give rise to dorsalization. So even at the ventral side, you can expect dorsalization. Alright, so manipulation Y, lithium chloride promotes dorsalization by activating the WNT signaling pathway, leading to excessive dorsal structure or even the formation of a secondary axis. Now what they are saying, the following statements were made with respect to the above manipulation and gene involved in setting up the dorsal ventral polarity in amphibian. The phenotype obtained due to manipulation X, that is this one, can be rescued by injection of noggin in one cell embryo. So this does make sense, because if you don't have cortical rotation, then beta catenin cannot give rise to dorsal structure. But if you directly inject the dorsalizing agent noggin, then yes, the phenotype can be rescued. You can have a dorsal structure also in this embryo. So A is correct. Okay. Chordin mRNA, chordin is also dorsalizing agent, will be enriched in embryos of manipulation X as compared with manipulation Y. This is not correct because, as I said, if cortical rotation is not there, you will not have the organizer, and if organizer is not there, noggin, chordin, anything will not be released from there. Okay, and if it is not released, then you cannot get dorsalization. So how can it be same as Y? Okay, injection of cDNA for chordin. So cDNA for chordin means it is you are injecting the chordin into ventral blastomere. So if you inject it towards the ventral blastomere, yes, chordin is a ventralizing agent, so it will lead to the induction of a secondary axis. So again, C is correct. And depletion of dorsalizing beta catenin transcripts in one cell embryo by antisense oligonucleotide leads to the phenotype similar to that obtained from manipulation X. If you deplete, means lower the beta catenin transcript in one cell embryo, okay, by using antisense, then you cannot expect the beta catenin to be present towards the dorsal side, which is same what happens in the embryo X. Okay, so even D is correct. The correct option over here is 1, C and D. This answer is correct. Okay. Now the last question over here, the cadherin-catenin complex is extremely important during compaction from a morula to a blastula. Transition of early embryonic cells into a blastula differed depending on the presence or absence of calcium ions. So we know that the cadherin and catenin, they form a complex together. So if you look over here, two cells, they can stay very close to each other. You have the catenin, beta catenin, and with the cadherin. So catenin is joining this cadherin, okay, and this cadherin is joining with another cadherin which is coming from another cell. So that's why this complex is very important, and you have this cadherin-catenin complex. It is necessary because compaction means in the morula, the cells are little bit loose connection, but in blastula, the cells get a very close connection. They become compact to each other. So adherence, the surface adherence of cells increases, because of which the cells come close to each other. Transition of early embryonic cells into a blastula, it will differ, like whether it is able to make into a blastula or not. It will depend on presence or absence of calcium ion. So let me tell you, cadherin, it depends on calcium, the binding adherence, it has calcium binding site. So an investigator blocked the expression of beta-catenin using vivo morpholinos to detect its effect simultaneously on compaction. So they were testing both for presence or absence of calcium, what effect was there, plus they were also checking whether blocking the expression of beta-catenin had any effect or not. So the answer is saying, which of the following condition will lead to the most successful transition of morula into blastula? The blastula will continue to develop even when calcium ions are present, since beta-catenin will get automatically activated by cadherins. Okay. So how can this be possible? How can beta-catenin, cadherins require calcium ions to function, and beta-catenin does not get activated automatically without the cadherin-catenin complex being formed. So without calcium ions, cadherins cannot mediate cell-to-cell addition, okay, which is essential for compaction. That is why answer key 1 cannot be the option. CSIR has given answer key option 1, but we had released the PDF before and it was mentioned that here option 4 seems to be the correct answer. I think this question can be challenged over here, because you yourself see the blastula will continue to develop even when calcium ions are absent. How? Cadherins will not join if calcium is absent. Okay. And if you are blocking the expression of beta-catenin, then how will it get activated automatically by cadherin? You are using in vivo morpholinos. So how can they be automatically activated? So not correct. Then second is, in the absence of calcium ion, calcium ionophores will assess development even when vivo morpholino to beta-catenin are induced. Again here also, if calcium is still present, cadherin will work, but without binding with beta-catenin, cadherins cannot join with each other. So in option 2, calcium ionophores can artificially increase intracellular calcium level, which might help cadherin function in the absence of external calcium. However, if beta-catenin is blocked, the cadherin-catenin complex cannot function properly, so development will still be impaired. And the third level is presence of calcium ion will enhance development when in vivo morpholinos to cadherin have been introduced to the early embryo. This is also not possible, because if cadherins are blocked, even if you add calcium ion, nothing is going to happen, because cadherin themselves are necessary for cell addition. Okay. So option 4, yeah. So sorry, I told you the other one. So calcium ions are essential for cadherin function, which is necessary for beta-catenin to link cadherins to the actin cytoskeleton, facilitating compaction. So this is the condition that most accurately describe the requirements. Okay, which is given this will not be. Thus, option 3 will not lead to successful development. Okay. But however, option 4 seems to be correct, because presence of calcium ions are essential for activation of cadherin and also beta-catenin, so that actin gets mobilized and compaction will continue. So this makes sense, because calcium ions are essential for cadherin function, which is necessary for beta-catenin to link cadherins to the actin cytoskeleton, facilitating compaction. This is the condition that most accurately describes the requirements for successful transition from morula to blastula. Okay. So that was all for the questions from Unit 5. There was one question in evening session of Unit 5 from metamorphosis and regeneration, which also did not seem to make any sense. So even that question, what I feel would be marked incorrect by CSIR. Okay, that's why I haven't discussed it in this session. So now students, I am giving the controls to Shamrita ma'am, so that ma'am can discuss the plant development questions with you. Okay, thank you. Good night.